Hello, my name is Vienna Buckles. I'm currently a third-year medical student at the University of Alberta. Our paper, Natural Green Spaces, Sensitization to Allergens and the Role of the Gut Microbiota During Infancy, follows infants from the Edmonton site of the Canadian Healthy Infant Longitudinal Development Cohort Study, or CHILD study. Allergic diseases, including asthma, are highly prevalent in Canada and place a considerable burden on families and the healthcare system. It is known that children growing up on farms have fewer allergies, potentially due to their healthier gut microbiomes. Thus, we are curious to examine relationships between infant exposure to natural green spaces in a Canadian city and future allergen sensitization, and also to determine whether this could be mediated by gut microbiota diversity at 4 months of age. In our analyses, we used a high-resolution topographical map of the City of Edmonton, Alberta, Canada, and the infant's postal codes at birth to quantify the degree of natural green space within 500 meters of the home. Fecal samples were collected at 4 months after birth and used to profile the gut microbiome. Additionally, infants were tested for atopic sensitizations to common food and inhalant allergens via skin prick testing at 1 and 3 years old. With these data, we perform logistic regression analyses to examine relationships between natural green space exposure and the development of single or multiple food or inhalant allergen sensitizations. After adjustment, we found there was a significant protective effect of having any green space within 500 meters of the home and having 2 or more inhalant allergen sensitizations at 3 years old. Additionally, we found that a significantly reduced symptom diversity of Actinobacteria mediated this protective effect. The lower values for the Actinobacteria symptom diversity measures observed in those infants with exposure to natural green space may indicate enrichment with a few dominant bifidobacteria, such as B. infantis. This microbiome profile is typically seen in breastfed infants. Thus, these findings are important for infants who are not breastfed for various reasons. Overall, our findings suggest that green space exposure can lead to bifidobacterial enrichment of the infant gut, and in turn, lower the rate of allergen sensitization and potentially of asthma development. The strengths and limitations of this study are further explored in the article. While further studies are needed to delve into the relationships between green space exposure and protective changes to the infant microbiome, this study provides a glimpse of how this relationship might look in the Canadian urban context. Thank you for your time, and on behalf of all of the authors, I hope you enjoyed the article.